Hello everyone. We have discussed several experimental designs. A standard experimental design comes with standard analysis. However, in practice, we often run several experiments. If these experiments have similar treatments, can we do a combined analysis across these experiments? Is there any advantage of doing the combined analysis? These are the aims of the lecture. In practice, we often run multiple experiments in the same field. Here is an example. This field is divided into 4 management practices, which are combinations of 2 irrigation types and 2 levels of nitrogen. There are multiple experiments in this field. Experiments with the same colors have the same treatments and the same research question. So, we can combine the data from the same colors experiments. In this example, we can combine the data from the 4 density experiment. This is the layout for a density experiment conducted in 4 different management practices. For each management, it is an RCBD with 4 replications. If we are interested in comparing the effect of management, a split plot or a split plot design could be a better option. There are 2 options to analyze the data from this density experiment. We can analyze the data from each management separately. However, we cannot evaluate the interaction between treatments and management. We also only have 4 observations to estimate the mean of each density treatment. We can do a combined analysis. However, the model would not be as simple as if these experiments were done as either a split plot or a split plot design. By combining the data, we can get a better estimate of the mean of the density treatment because we now use 16 observations to estimate the mean of each density treatment. We can do a combined analysis by either assuming the homogeneity of variance, so we pool the residual variance across these 4 experiments, or by relaxing this homogeneity assumption and estimating the residual variance for each experiment. Let's assume that these 4 experiments have similar residual variances so that we can use the pool residual variance. There are 2 models for the combined analysis. Model 1, we analyze this data as a 2-factor experiment of management and density, where management is combination of irrigation types and nitrogen levels. The skeleton ANOVA. Block is nested within management because block 1 in management 1 is not the same as block 1 in management 2, and etc. Model 2, we analyze this data as a 3-factor experiment of irrigation, nitrogen, and density. In this model, we can evaluate the interaction between irrigation and nitrogen. The skeleton ANOVA. Before doing the analysis, we should check the data by running some preliminary data analysis. If the rows and columns are recorded, we can create heat map to check the experimental layout. From this heat map, we can see that there is a misalignment of column in one of the replication that due to a mistake during planting. We can also see that the treatments are properly randomized. There is no pair of treatments that always occur together. It seems there is no spatial trend on the dry weight. We can do box plot to display the summary statistics. Based on this box plot, we can see that there are no outliers in the data. This box plot also indicates that the interaction between management and density might not be significant, as the ranking of the density are the same across management. This is the ANOVA table for Model 1. Based on this table, we can conclude that there is no evidence of significant interaction between density and management at the 5% significant level. Because there is no significant interaction, we can conclude that the main effect of management and density are both significant. This is the ANOVA table for Model 2. At the 5% significance level, we can conclude that the highest order interaction of density by nitrogen by irrigation and the second order interactions of density by nitrogen and density by irrigation are not significant. So we can conclude that there is a significance effect of density. The second order interaction of nitrogen by irrigation is significant. So we cannot make any conclusion about the main effect of nitrogen and irrigation. Combining similar experiments could increase the power of the analysis. However, we must carefully think about the model we are going to use to analyze the data. Now, can you do this?